this study, and I'm pretty sure I can do her justice. But if you do have questions, do reach out to Holland, because this is certainly her baby. So this subject touches more on what I call like the core academic focus of my lab, which is thinking about what really cool biology can we learn from coral in the context of restoration, because I see restoration as like really awesome in an applied sense, but also it's like this perfect experimental system from a biological standpoint. It's almost like you restoration practitioners are creating these like lab rats, essentially, right? Like in the human study, there's all these facilities that generate lines of lab rats that you can always go back to that same line of rat or mouse and like do the same studies and learn a lot about these different genotypes. And now with restoration, like we have the capacity to do that as well. And one of the things my lab's really interested in is the idea of plasticity, or in the simplest sense, like the ability for one genotype to change its phenotype or change its physiology in response to an environmental stimulus. And we see this in acroporids. Like these, we can see plasticity just going into the field, right? If you go and look, in this case, Acropora Palmata will take on these really incredible morphologies in response to the different environmental pressures. And we're really curious about how this influence of the environment might interact with different genetic lines to influence the overall performance characteristics of coral. So why would we care about G by E in the context of restoration? Well, as you've heard about in some of the cool talks that preceded this break, plasticity is really important for understanding how coral are going to respond to the different environments in terms of like predicting the impacts of your restoration. But also, if you overlay, say, plasticity in a single trait, so this is, each of these lines would be a genotype. This genotype is changing in response to whatever environmental axis of variation. Knowing something about how that change in trait value overlays on fitness can tell you something about the performance characteristics of that coral. And by fitness, I mean survival, you live, or reproduction, you reproduce. And so if you can understand how that change in trait value is functional, you might see that your plasticity is adaptive, or it benefits you, right? You have an ecological response and that's actually helping you. Plasticity can also be maladaptive. You might have a change, but it actually like sucks, right? You're on your way out in terms of having that happen. Or it can just be neutral, right? It might be that you see this big change in trait value, but it doesn't actually impact the overall survival or reproduction of your favorite organism. And so we want to know, how is plasticity linked to these other characteristics? And can we use that information maybe to try to optimize genotype and site selection going forward? So the main questions for this study in Palmata was, do you see genotype by environment impacts on the growth and survival of Acropora palmata outplants? We didn't look at reproduction. Survival was like our fitness metric in this case. And how do these changes impact like the community of Acropora palmata? And we think about community here in this context of sort of like other organisms that might be living on or around. So in this case, we looked at microbial communities, so bacterial communities, as well as the communities of symbionts that these corals house, and found some different things. So to look at growth, we used individual colony photogrammetry. So you might have seen all these beautiful like reefscape images. In my lab, we've taken that and gotten really small and just done it for individual coral colonies, because it's a really powerful way to get lots of complex information from a single colony. And those protocols are published and available on Protocols.io if you want to do it yourselves going forward. So what it is we just take a whole bunch of photos of an individual colony, turn that into a three-dimensional model, and then instead of sitting there with a little like bendy ruler on the reef trying to measure everything, we just take our time and measure things like, yes, growth, but also surface area, volume, and the volume of the interstitial space, which is what I think of as the negative space created by the branching of that colony, right? Like as the colony grows, literally between its branches is what the habitat is for fish. So that's what interstitial space is. And then we did the classic ITS2 types, amplicon sequencing, and then amplicon sequencing for the 16S, which is a marker to look at bacterial community diversity. We did this in the context of a pretty big transplant experiment facilitated by a collaboration with Moat Marine Lab. So they were able to give us 5 different genotypes of their sexually produced Acropora Palmata, so represented here by like the 5 different colors. What we did was we got 3 replicates of each of those genotypes, so 3 ramets, in this case like 3 little plugs of the sexually produced offspring, so 3 clonal replicates of each genotype that we outplanted to each of 9 sites along the lower Florida Keys, represented by each of these circles. We picked these sites specifically because they overlapped with fixed water quality monitoring stations for the Keys. I'm not going to talk about that data today, but it allows us to get a little more holistic information about what's potentially influencing differences in performance. I think that's all I want to say. Yeah, and these are all her notes on that. Cool. So a little backstory about these particular ramets. As you know, Moat's been doing a really incredible amount of sexual reproduction in their restoration program. And so what they do is they take individual larvae, those are settled, they start to grow. Once they get big enough, they start to microfrag those. And those are all maintained in their land-based nursery system. So what I was particularly excited about with these guys was, oh wow, we've like perfectly controlled environment for their entire developmental history. So not only do I have like perfectly controlled genotype, that prior conditioning environment, which is really hard to get at when you collect corals at random from the field, is also controlled when you have this beautiful like land-based nursery rearing system where someone else has been like rotating them and caring for them so gently. And they were raised in those conditions for like 4 to 5 years. So even though they were individually like quite small, age-wise, they were actually like fairly old when you think about it. And we put them out in the field. We let them go for 2 years. We took photographs quarterly, so every 3 months, and then tracked survival and growth, as I said. And at the end of the 2-year time point, we took tissue samples. So we took samples initially and then at the end to quantify those other performance metrics. What did we find? Something that surprised me. So for survival, we actually didn't see any effects of genotype. So this is showing you a survival curve, so it's just plotting starting out. Everybody's at 100% survival at the start of the experiment, and then how many individuals died at each time point. And you can see survival actually maintained pretty high after that 2-year outplant time point, about 75% overall on average, but it wasn't different across genotypes. We just saw similar survival. When you look at site, however, there were like massive effects. Before we even got to the 2-year time point, all of our coral at Bahia Honda were dead, toast. Whereas we saw some sites that still had like 100% survival. Eastern Sambo was like, whoa, amazing. Everybody survived there. So massive side effects. The other thing we did see though for survival, which we're still trying to scratch our head around, is that even though there wasn't necessarily an effective genotype, there was an interaction between genotype and site in the sense that not the same genotype survived or like exhibited the highest survival as you went across sites. So even though on average that all canceled out, if you looked at it site by site, it was different genotypes that were actually surviving at each site, which is represented by this. What this is a correlation matrix. So it's saying for each pairwise comparison of sites, how similar are the rankings in terms of survival across the genotypes? And blue would be a strong positive correlation, like it's always the same that are surviving. So like Eastern Sambo, Lukey, we see the same genotypes surviving at each of those. Red is actually a strong negative correlation, like it's totally not predictable. Different ones are surviving at each combination of sites, even though on average they're all doing okay. And again, this is just another representation of thinking about the differences in growth. So this is a crazy way to look at it. We also see site as the primary factor affecting growth. So this is growth rate on the y-axis. We're looking at it, I think this is actually for surface area, but it was similar for the other, yes, this is surface area. It was similar for the other traits. I'm just going to show you surface area here. But you can see that if you look at mean growth of all the genotypes across site, fairly low. At Big Pine, much higher at Eastern Dry Rocks. And these individual lines are just tracking for those individual genotypes going across the plot. So site was still the primary driver for growth in our data set as well. So really big side effects for Palmata. When we looked at the microbial communities, we didn't actually see any correlations here or big drivers. So this is just a way of visualizing overall differences as a function of the different factors in our study. So it's thinking about the diversity of the microbial communities overall. And I'm mainly just showing this to illustrate what we found that was the biggest driver in our data set was actually just time point. So whether we took microbiome samples from the coral in the nursery raceways versus when they went out to these different field sites. But when you split it up, there's, like, even looking at it colored, there's not actually big clustering necessarily by genotype or by different sites, right? They're not like splitting out as like, oh, this one was really different or that one was really different, even though we saw those big site differences in growth and survival. So large effective time and also condition, right? Because these were taken from the raceway versus the outplant sites, no effective genotype. The story's different for ITS2, and I'd love to hear folks' thoughts on this because this one also really has us scratching our heads here. So this is looking at a similar distribution, but now we're thinking about the algal symbiont communities in these coral. And for most of them, we see this really funny pattern where it's like, okay, they're all clumping together. They all look very similar, except for this funny batch right here, which is one of our genotypes from the raceway. They sort of cluster out separately. And then we have all of these other just like random individual dots that are like, wow, these are really funny outliers. When you look at this, what happened in the raceways, you might have already seen this with some of the other talks from Moat. The land-based nursery selects pretty strongly for a non-standard symbiont type in the Moat corals. So they are all pretty much dominated by Duristinium. And then except for that 13XK, which showed this co-infection with cladocopium, which we've actually seen from Shelby's talk, was like, okay, cool, we're not the only ones seeing this kind of a pattern. So this is 10 replicate ramets. Like we randomly sampled 10 replicate ramets prior to outplanting from the nursery. 100% Duristinium, except for this one really weird signature of the co-infection in that genotype, super consistent across all the ramets. So that, like, everybody's Duristinium. Why they're all clustering together 2 years later, most of them stay Duristinium in the field. They keep that Duristinium, except for these few weirdos that actually do switch back to Phytae. So they switch back to, like, or I don't know if it's shuffling, switching, they switch back to the dominant site. We never detected Phytae in any of the raceway corals. We only see it in these field transplants, and what was crazy is you can't see the blues here, but we actually get a couple different ITS type profiles, and it's actually site specific. So like certain sites tend to have different ones, which makes us think possibly they're acquiring this Phytae once they're getting out to the field. But what I can't figure out is, one, right, if in natural populations, like they associate with Phytae so closely, why is it that when given the chance to take up Phytae, they don't all do it? They don't all do it. Two, we never saw bleaching. We monitored these guys every 3 months for 2 years. They did not bleach. In fact, secretly, I was hoping there would be a bleaching because I was like, oh, this would be really cool to look at in the context of our G/E experiment. Did not happen. And yet, we still saw complete turnover of the symbiont community. How the heck did they do that? I don't know. So if anybody has any ideas, would love to hear it. So yeah, that's that. So what we saw is we don't see any site or genotype effects for the microbiome. We don't see major site or genotype effects for the symbiont community. That's because most of them keep Duristinium, even though there were those weird few instances of shuffling. We don't see any effect. We don't see any evidence for plasticity among differences like G/E, although we do see this big site effect. So an average effect of responsiveness, not like differences among genotypes, I should say, in their plasticity. Although we do see genotype by environment interactions for survival. And so with that, Holland's picture is not on here. It should be, because this is her talk. So Holland's been leading all of this work, and yeah, we have a great set of students and collaborators that helped. Thank you.
Thank you. I think we can take one quick question while we're just transitioning slides over to anyone has a burning question they want to ask Colin.
Yes.
Super cool that you were able to use all separate